All right, let's get started. So, first of all, congratulations, you made it to the last week of class. So, that being said, few reminders. What is due this week? Thursday, if you're doing quiz 12, let's do Thursday night. And Friday, your projects are due. So, if you have not finished yet, finish by Friday. Any questions at all about any of those assignments that are due? Your final exam is the following Thursday, the 14th. It'll be in this room. It's at 12 o'clock noon, so our normal class starts at 12:30. Make sure you get here at noon on Thursday the 14th for the third exam. Office hours and everything are posted for this week and next week. They're a little different than normal, so if you want to come see me, make sure you check on Brightspace for my office hours. You can always send me an email. So yeah, that's all of the logistical stuff I want to talk about. So let's get into chapter 17. So chapter 17 is about the citric acid cycle. But before we can get to the citric acid cycle, we sort of orient ourselves with where we left off in glycolysis. So with glycolysis, we turned glucose into pyruvate. So now we're at this point where we want to enter the citric acid cycle, but to enter the citric acid cycle, we don't need pyruvate, we need acetyl-CoA. So the step in between to help us get from pyruvate to acetyl-CoA is going to be our pyruvate dehydrogenase complex. So we are going to go over the PDC first, because that is the very important middle step. So first of all, let's discuss the overall reaction that's catalyzed by the PDC. So can anybody tell me any reactants or any products? And so recall for this part, when I ask you questions like this, we want to track the carbons that are originally from glucose. What are they going from to? And then we want to track energy, and then we want to track electron carriers. So yeah, you want to give us some starting points? Yeah, very important. Pyruvate. So acetyl-CoA has 2 carbons. Yeah, so we have, yes. Exactly, yeah. So pyruvate, the 3 carbons, 2 of them go to the acetyl group, 1 of them goes to a carbon dioxide. Yeah, so let's start from there. So we have pyruvate. Acetyl-CoA has 2 carbons. The 3rd carbon is a carbon dioxide. And if we have CoA as a reactant, what do we now need as a product? Or other way around. If we have CoA as a product, what do we need as a reactant? CoA, yeah. So that's another one. Coenzyme A. And then what else do we need in terms of energy or electron carriers? Yeah. Fabulous, yeah. So we have an NAD plus going in. We have NADH coming out. So, and this makes sense, right? We're oxidizing our carbons from our pyruvate, which I spelled wrong, my bad. And in turn, we're reducing our NAD. Let me change my typo. Can't talk and write at the same time. Pyruvate. Okay. Great. So, how does the PDC fit into glucose metabolism overall? So we kind of just talked about it before we got started. Yeah. Yeah. Yeah, so it's the bridge between glycolysis and what? I really am struggling with letters today. So before we get to the electron transport chain, there is another step. So we still have 2 more carbons that have to get oxidized down. In what? Yeah, the citric acid cycle, yeah. So Yeah, so we're going to be calling a lot of these things by letter. So the PDC, that's the pyruvate dehydrogenase complex. The CAC, that's the citric acid cycle. So the PDC helps bring us from our pyruvate that came from glycolysis into our acetyl-CoA that we need for the citric acid cycle, which is what we see in our overall reaction. And what conditions do we need starting now with the PDC in terms of like the environment? Yeah. I'm sorry? Low energy. Yes, that's true. So yeah, we're using this as a way to make more energy. That is true. But I'm thinking there's one very important thing that we need in the environment to go through with cellular respiration. Yeah. Oxygen, yeah. So from this point forward, oxygen is required. This is aerobic. And we talked about in the video how if you don't have oxygen, you sort of start to get backed up at this point. You start to build up the products like NADH. And so as a result, these reactions won't move forward at a certain point anymore if you lack oxygen. That helps to regenerate NADH back to NAD+. Because the NADH is what we bring forward into the electron transport chain to ultimately help us make ATP that we'll talk about on Thursday. And so if you can't get rid of that because you lack oxygen, then you end up building up all this product, all this NADH, and the reaction will no longer go forward. So that might bring up the question of what happens when there isn't enough oxygen. So during aerobic conditions, when we have enough oxygen, this is what we're going to talk about today. The pyruvate dehydrogenase complex and the citric acid cycle will both happen. And that's shown on the left side of this diagram. This is aerobic conditions. The glucose, all of the carbons from our starting material, which is glucose, get fully oxidized into carbon dioxide. Water is the final, oxygen is the final electron acceptor, and that forms water ultimately. And so this is also great because this whole process, including what we'll talk about on Thursday, the electron transport chain and the ATP synthase, help us make a lot of ATP. So every glucose molecule can yield over 30 ATP this way. In anaerobic conditions, when there's a lack of oxygen, we can't do this, right? Because we have this buildup of products, and so the reactions won't go forward anymore. So we have some ways to get around that. So during anaerobic conditions, we do what's called fermentation. And that is different depending on what type of organism you are. So in humans, organisms like us, we do what's called homolactic fermentation, which is shown in this middle arrow here. Homolactic fermentation takes pyruvate and turns it into lactate. And the reason why this is really important is actually because of our electron carrier. So what's happening when pyruvate gets reduced into lactate is that NADH gets reoxidized into NAD+. And now there's enough more NAD+ that's available to go back through glycolysis, because glycolysis can happen under anaerobic conditions, and it gives us a little bit of ATP. Every glucose gives us 2 ATPs during glycolysis. So fermentation in low oxygen conditions can help us make a little bit of ATP through the process of regenerating that NAD+. So, yeah, and like I mentioned in humans, that's called homolactic fermentation. Does anybody have any idea what enzyme might do this that turns pyruvate into lactate while it oxidizes NADH to NAD+? Yeah. Yeah, LDH, lactate dehydrogenase. So if you're in the lab class for this course, we spent about 5 weeks working with this wonderful enzyme, LDH. And this is what it does. It's a really important part of metabolism under anaerobic conditions. So yeah, so that's humans. For other types of organisms like yeast, they go through a different type of fermentation called alcoholic fermentation. That's what we see on the right in this diagram, where the pyruvate gets oxidized into ethanol. But it has the same result, which is regenerating more NAD+ that they can use for glycolysis. So fermentation, sort of regardless of what you do with the carbons, the sort of important part is what you're doing with your electron carriers. And like we mentioned, this can let you do glycolysis and help you get at least 2 ATPs for every glucose. And then in people, so we talked about how in people we make lactate. We have a process called the Cori cycle. The Cori cycle is when we can transport lactate to the liver and then use that lactate to help regenerate more glucose through gluconeogenesis that can ultimately be used in glycolysis. And so this is a really important thing. So we have sort of one central molecule, pyruvate, that depending on what's going on in the environment can have a very different fate and yield very different amounts of energy. Any questions about that? All right. So that is really all I had to say about the pyruvate dehydrogenase complex. So we had pyruvate. When there's oxygen, we were able to successfully turn it into acetyl-CoA. Now we're ready for the citric acid cycle. So let's once again do our overall reaction with the citric acid cycle. So can anybody throw out some reactants and products in terms of where our carbon's going, where's our energy going, where are our electron carriers going? Yeah. Yeah, we make 3-NADH. That is definitely one piece. So if we're making 3-NADH on the product side, what do we need on the reactant side? Yeah. 3-NAD+. Yep. What else do we need? Yeah. Yes. Yeah, so we started with our acetyl-CoA that we made with our PDC, the 2 carbons. So now these carbons are finally getting fully oxidized all the way to carbon dioxide. And we free up that CoA to be reused next time. Anything else that we need? Yeah. Yeah, so we have some energy currency that we directly make in the form of GTP. So we need GDP plus inorganic phosphate on the left, and on the right we make 1 GTP for every turn of the citric acid cycle. And there is one more thing that I'm thinking of. Yeah. Yeah. Yeah, how many? Just 1. Yeah, awesome. So that's our other electron carrier. Yeah, so this pretty much accounts for all the interesting stuff. Where are our carbons going? Where are our electron carriers going? And are we making any energy currency directly? Okay, so, yeah. Yes, sorry, I forgot to. I even wrote 2 carbons there to remind myself, and then I forgot to write 2 carbon dioxides. That's true, yeah. Thank you. Any other questions or math errors? Yes. Yeah, for every glucose, this goes twice, exactly, because every glucose makes 2 pyruvates, and each pyruvate makes 1 acetyl-CoA. So, yeah, double it for 1 glucose molecule, exactly. And we'll go through that later in this, actually on the next slide or so, yeah. Cool. Okay, so let's start talking about some of the specific steps of the citric acid cycle. So what molecule is consumed in the first step and regenerated in the last step of the citric acid cycle? Yeah, oxaloacetate. And a lot of times you'll also see this written as OAA, so you can feel free to use that as shorthand. That's short for oxaloacetic acid, which is essentially the same thing as oxaloacetate. Does anybody remember where else we've seen oxaloacetate in the past week or two? Yeah. Yeah, which stuff? Exactly, yeah. So we saw oxaloacetate before when we talked about gluconeogenesis, when we reversed the 10-step of glycolysis. We have that two-step reaction in gluconeogenesis, and that first step is turning pyruvate directly into oxaloacetate. And so that's the same molecule. So that molecule can be used in gluconeogenesis, and it can also be used to help fuel the citric acid cycle. And oxaloacetate is a really helpful intermediate that if you learn more metabolism for other types of macromolecules, you will see popping up again as well. It's a really good way for how our bodies can integrate protein and lipid metabolism along with glucose metabolism and sort of pick and choose where everything is going. Okay, so yeah, how does the citric acid cycle fit into glucose metabolism? Yeah. Yeah, exactly. Sort of, so it follows the PDC. Prepares us for the electron transport chain. So what are the important things that are happening during the citric acid cycle that are sort of helping us get ready for oxidative phosphorylation in the electron transport chain? Yeah.
You're creating proton donors?
Yeah, I mean, we're making reduced electron carriers is a helpful, yeah. So we're making lots of reduced electron carriers. Which will donate both electrons and protons. And then what other sort of exciting thing is happening during the citric acid cycle with our carbons that came from glucose? It's not that exciting, I guess. So at this point, all of our carbons are fully oxidized. We have every single one of our 6 carbons from glucose, but at the end of the citric acid cycle, they have all turned into carbon dioxide. So yeah, all of our carbons are now oxidized. A lot of our electrons are now hanging out with the electron carriers, and they're ready for sort of the last part where we actually do oxidative phosphorylation. Any questions about the citric acid cycle in general? All right. So let's start thinking about where is the energy going and coming from. So here's a little table. We're looking at the pyruvate dehydrogenate complex and the citric acid cycle. So I want everyone to take a few minutes and start filling in for each of these processes from that chapter 17. How many GTPs are made per cycle, how many NADH, how many FADH2, and how much carbon dioxide. I just want to, before you get started and doing math, per cycle means for every pyruvate that goes in. And whenever I ask you math questions like this, I'll always say per cycle or per glucose, so it's clear how many you had to be thinking about. So take a couple of minutes and do some math. Alright, so let's start with the PDC. So how many GTPs are made for every cycle in the PDC? Yeah, none. Yeah. How about NADH? 1. How about FADH2? 0. How about carbon dioxide? Yeah. 1. Yeah, so at this point we have 1 pyruvate, we made it into an acetyl group, so we lost 1 carbon dioxide. So how about the citric acid cycle? How many GTPs? Yeah. 1 NADHs. Yeah. 3. 1 and 2. Yeah. So yeah, so this is basically summarizing everything we talked about on the last slide. So we wrote out the full reaction, and this is kind of breaking out the reaction into just tracking the important aspects in terms of energy. So, and so we just talked about how per cycle means for every 1 pyruvate. So how would this change if this was actually for every glucose molecule? What would all these numbers do? Yeah, they would double. Yeah. So we don't have to go through doubling them, but this is all of them doubled, right? You have PDC from instead of 1 NADH, you'd have 2. Instead of 1 CO2, you'd have 2. For the citric acid cycle, for every glucose, you get 2 GTPs, 6 NADHs, 2 FADH2s, and 4 carbon dioxides. So, yeah.
So you said that per cycle means 1 pyruvate?
Yeah, so for the citric acid cycle, if you just think about the citric acid cycle, you're starting out with 1 acetyl-CoA enters, and you go around 1 turn of the cycle. That's 1 cycle of the citric acid cycle. So 1 pyruvate. Yeah. So just out of curiosity, let's also fill this in for glycolysis, just so we can start thinking about kind of all the processes we've done so far. So so in glycolysis we didn't make GTPs. What energy carrier did we make? ATP. And how many did we make for glucose? 2. How many NADHs did we make? Also 2. How about FADH2s? None. And how about CO2s? What's that? Yeah, also none. Yeah. Yeah, so for every glucose, 2 of the there's 6 carbons in glucose, right? So 2 of them get carbon dioxide in the PDC, and the other 4 are in the citric acid cycle. So glycolysis gets you going, but doesn't fully oxidize any of the carbons yet, just in glycolysis. So if we start thinking about adding up these total amounts of energy that we have so far, so how many sort of ATP equivalents do we get for every glucose so far? 4, yeah. And NADHs, we get 2 plus 6 plus 2 is 10. For FADH2s, we get 2. And of course, total carbon dioxides, we should have 6. So these are the sorts of energy totals you can start to keep in mind, especially on Thursday once we start tracking the energy that we get out of all of the NADHs and FADH2s. These are all numbers you'll have to be very comfortable working with. Any questions on where any of these numbers came from or anything else? Yeah. And adding up per glucose, how many total ATP equivalents, how many NADHs, how many FADH2s, how many CO2s? Yeah. Per glucose. All right. So let's start talking more about the actual citric acid cycle. So I'd like everyone to take a few minutes and start putting the cycle in order. For now, feel free to look back at your notes, but just as for the other pathways on the exam, you should be able to fill in a completely blank pathway with all of the enzymes and all the metabolites and all of the little extra bits and pieces. So you can take a few minutes now and start to put these in order, and then we'll talk about them together. All right, let's see if we can put them all in order. So which enzyme comes first? Yeah. Citrate synthase. Yeah, it's a tongue twister. What is the next one? Yeah, aconditase. How about the next one? Go ahead. Isocitrate dehydrogenase. All right, what's next? Yeah. Yeah, alpha-ketoglutarate dehydrogenase. It's mostly just fun to say all of these. What is the next one? Yeah. Succinyl-CoA synthetase. And what do we have next? Yeah. Succinate dehydrogenase. All right, we got 2 left. Yeah. Fumarase. And what's our last one? Yeah. Malate dehydrogenase. All right, so that was a lot of words that probably don't sound like they mean a lot. So let's go through what each of these enzymes actually does. So we can start from the beginning. We have citrate synthase. So what are the reactants that go into our reaction that citrate synthase catalyzes? Yeah. Yeah. Uh-huh. Yeah. So For now, I'll probably ignore stuff like water, but know that they go in. But just like sort of just for tracking carbons purposes and energy purposes is sort of what we're going to focus on here. So for citrate synthase, we have what's going in, our acetyl-CoA. This comes from the pyruvate dehydrogenate complex that just helped us make this, very important as our input. This one has 2 carbons, and it's getting combined together with oxaloacetate. Or OAA. Oxaloacetate has how many carbons? 4 carbons. And so, yeah, and keep in mind oxaloacetate, this is a cycle, right? So we start with oxaloacetate. Spoiler alert, we're going to finish with oxaloacetate. So that's something that can help you remember, maybe. So those are the 2 big things that are going in. And then what's coming out? Citrate. Yeah, and how many carbons does citrate have? Yeah, so citrate has 6 carbons. So we glued together all of our carbons. And what's left over? Yeah, CoA, exactly. So we took the 2 carbons off the CoA, we stuck them onto the oxaloacetate, we made a citrate and a CoA. What kind of enzyme is this that takes a piece off of one substrate and puts it on the other substrate? It's a transferase, yeah. Okay, so that's our first one. What about our second one? Yeah. Yes. They're essentially both the same thing. Yeah, so when it's written as CoA-SH, that's just to remind you that it's the reduced form. So coenzyme A is important, especially here, because it forms this thioester bond. That's really high energy, so when you break it, it helps release energy, which helps to drive the reaction forward. When it's acetyl-CoA, there's the thioester bond is there. When it's broken, the CoA is reduced, and there's nothing attached to it. So it's just kind of saying like there's a hydrogen there instead of an acetyl group. You can write it that way. You can write it either way. They're both correct. Yeah. Yep. Any other questions? Okay, so so this is a cycle. So we're always going to be taking the product from one step. It's going to be the reactant in our next step. So what are we taking forward to our next step with aconitase? Yeah, citrate. So citrate has 6 carbons. And what's happening to it in this step? Yeah. Yeah, so it's turning into isocitrate. Exactly, yeah, so isocitrate also has 6 carbons. We start and end with the same atoms in the molecule. They're just rearranged. They're bonded together differently. So this enzyme is an isomerase. It's just rearranging the atoms within the substrate. All right, so that brings us to step 3, isocitrate dehydrogenase. So we're taking our isocitrate that we just made, like the name suggests, isocitrate dehydrogenase. What are we doing with isocitrate? Yeah. Yeah, so the isocitrate is getting oxidized into, do we know our product? Yeah, alpha-ketoglutarate, which you can absolutely write as alpha-Kg, because alpha-ketoglutarate is a very long word. So the isocitrate is turning into alpha-Kg. How many carbons does alpha-Kg have? 5. So what happened to our other carbon? Yeah, so this is our first carbon dioxide. And so remember, we started out, we brought 2 carbons in with us. So this is our first one. Our first one is now fully oxidized. We're done with it. What else is happening if we're oxidizing one of our carbons? Yeah. Yeah, we had to reduce our electron carrier. So we have now our NAD+, and it's reducing to NADH. So what kind of enzyme is this? Yeah, it's an oxidoreductase. Yeah, we're having the reduction of an electron carrier while we oxidize our carbon. All right, so that brings us to our next enzyme, which is alpha-ketoglutarate dehydrogenase. So these are both the same kind of enzyme, right? They're both dehydrogenases. So we have alpha-ketoglutarate, which has 5 carbons. What's happening to it? Yeah. Yeah, it gets made into succinyl-CoA. I need to move my arrow over. So how many carbons are in our succinyl group? 4 carbons, yeah. So what happened to our 5th carbon? Yeah, it's fully oxidized into carbon dioxide. So at this point in the citric acid cycle, we're only 4 steps in, we already fully oxidized both of our carbons. It's very overachiever. It's only halfway through, already fully oxidized. So after this reaction, the other 4 steps are all about just coming back to our starting molecule, which was oxaloacetate, and also helping us generate more energy in the following steps by reducing more electron carriers. But at this point, our carbons are essentially taken care of. But first, we had to finish out this step 4. So we know what happened to our carbons. What's happening in terms of, or actually, we have one other thing we have to add as a reactant, because we have CoA as a product. So what do we need as a reactant? Yes, so we need a regular CoA, or a CoA-SH, which you can also write it that way. And then what about electron carriers? Yeah. Yeah, and we have another NAD+, getting reduced to NADH. So what kind of enzyme is this one? Also an oxidoreductase. Yeah, so it works just like our previous step, and they have actually very similar outcomes where each of them ends up with 1 fully oxidized carbon and 1 NADH. And now we're back down to 4 carbons, and those 4 carbons are, like I just mentioned, going to get turned back into oxaloacetate over the next 4 steps. So that brings us to the 5th step. So we take our succinyl-CoA that we just made, and at this point I'm going to stop counting carbons because they're all 4 from now on. So what is happening here with our succinyl-CoA? Yeah, so it's getting turned into succinate, so we're separating it out from the CoA. And we just talked about how the thioester bond in the CoA is very high energy. So what else is happening at this point when we're breaking our bond to CoA? What are we making? Yeah. Yeah, exactly. So now we're doing a substrate level phosphorylation with all that energy we just released to make 1 molecule of GTP. And since we made GTP, that means we had to put in GDP and an inorganic phosphate. So what kind of what kind of enzyme is this one? Yeah. It is not a transferase because we're not moving something from a substrate to another one. Yeah. It's also not an isomerase. It actually helps to think of it almost in the reverse direction. if that gives you a clue. So in the reverse direction, we're using, it looks like GTP hydrolysis to drive something else, right? So what kind of enzyme uses the hydrolysis of a high energy bond to bring together two substrates? A ligase, yeah. Yeah, so in the reverse direction, GTP is getting hydrolyzed to GDP, and succinate and CoA are getting stuck together into succinyl CoA. So it is a ligase. All right, so that brings us to number 6. So we made our succinate, so we bring that with us. And what is happening here? Yeah. Yeah, it's made into fumarate. And this is a dehydrogenase enzyme, so that should give you a clue for something else that's happening at the same time. Yeah, which electron carrier is it this time? Yes, so now we have our reduced electron carrier, FAD, or FAD is getting reduced, I should say, to FADH2, while succinate is getting oxidized to fumarate. And what kind of enzyme is this? Yeah. Yeah, it's an oxidoreductase. Yeah, so anytime you see dehydrogenase in the enzyme name, it's an oxidoreductase. And it's always going to have an electron carrier that's getting oxidized or reduced, depending which direction you're going. Okay, so now we're getting there. We got 2 steps left. So number 7, we have fumarase. So we take our fumarate that we just made, and what's happening here? Yeah. Yeah.
So for succinate,
Yeah.
I was confused because it showed there was a double bond forming.
Okay, let's go look at the picture. So many pictures. Yes, so there is a double bond because you're removing these 2 hydrogens. It's a dehydrogenase. So you're taking out the hydrogens and their electrons to help reduce the electron carrier. So yeah, I guess. Okay. Yeah, I was trying to think of it's different from a lyase because for a lyase, when you remove the pieces, they go by themselves. They're not you're not taking those electrons and sticking them onto your electron carrier.
Yeah.
If that kind of helps. Okay. Yeah. Yeah, essentially, because you're taking away bonds to hydrogen, so you're oxidizing them. Okay. Yeah, some of them are trickier than others, for sure. I gotta go back through all these. Oh boy, no. Okay. So let's go to our fumarate. So what is fumarate getting made into using fumarase? Yeah. Malate, yeah. And if it sort of helps, the naming for each of these is also in each of the following enzymes. So we made fumarate in the last step. Now fumarase is going to do something with fumarate and make it into malate, which is named in the enzyme in the next step. So in a lot of ways, these enzymes are kind of named for their reverse reaction, which is a little bit confusing, but the names at least are in a pattern of a sort. And so what's happening here when we're making fumarate into malate? What are we adding? Yeah, water. Yeah, so what kind of enzyme is this one? Yeah. So yeah, this one's also a little bit of a tricky one. So let's go look at the picture where we just were. Okay. So we are, so it's not really a transferase because we're not taking a group from a substrate and pulling it off of there to another one. We're taking the whole thing and putting it in there. This is another one where it helps to think about the reverse reaction. So if we're pulling off these two pieces and forming a double bond, yeah, so this one is a lyase. And then it's becoming water as the second product. And it's it's a lyase. You can also, this is also a hydration because you're adding water. But remember, it's not hydrolysis, right? We're not using water to break a bond and make two separate products. Okay, so that brings us to our last step. So our last step, we take our malate. We're using malate dehydrogenase. So we should all know what the product is. We're regenerating the very first molecule that we started with, which was oxaloacetate. And this is a dehydrogenase. So what do we need to do at the same time? Yes. Which electron carrier are we reducing? Yeah. We're going to take our NAD+. We're going to make NADH. So I know it's a lot, but hopefully you can sort of see how each one is a logical progression. You can get a lot of information from the names of the enzymes and the names of the metabolites that are being created. So of these 8 enzymes, which ones are the regulated steps? Yeah. Yeah, citrate synthase is one. Sorry, I'm just changing colors here. Yes, isocitrate dehydrogenase. And yeah, alpha KG Tired of saying words. Yeah. So these 3 that I just circled are the regulated ones. So these ones also happen to be, especially 3 and 4, these are the ones where we're releasing our final 2 oxidized carbons. The first step is also important to regulate because once you start the cycle, it's going to kind of have to go through the whole thing. Yeah, and also these 2 steps, the 3rd and 4th step at least, where we're reducing electron carriers, these can also be places where you might get stuck in a low oxygen situation. So also kind of makes sense that these are regulated steps that are important for deciding whether or not we want to go through with the whole citric acid cycle. Any questions about the citric acid cycle? All right. So that brings us to regulation of the citric acid cycle and the pyruvate dehydrogenase complex. So first we'll just review, or I'll have you all take a moment to review your notes. So just think about what is going to happen to the rate of flux through the 2 complexes, or the PDC and the CAC, in each of these 4 situations. So just think about whether it's going to increase or going to decrease. So if it goes faster or goes slower. And I'll give you a few minutes to think about it, and then we'll come together and talk about it. Yeah. All right, so let's keep going. So what's going to happen to the rate of flux through the PDC and the CAC when insulin gets released by the pancreas? Yeah. Yeah, it increases them both. So yeah, so why does that happen? Yeah, that's sort of like a more direct regulation. I mean, one other way you can think about it that's maybe more simple is insulin is a sign that you have high blood sugar. When you have high blood sugar, that means you have lots of glucose, which is your starting material for glycolysis. You go through glycolysis, you make a whole lot of pyruvate. If you have a whole lot of pyruvate, then the PDC is going to help you make a whole bunch of acetyl-CoA. And if you have a whole bunch of acetyl-CoA, then you have lots of starting material for the CAC. So a lot of this is the way you can think about is, do I have lots of reactants or do I have lots of products? So when you have insulin release, that means you have lots of reactants. So how about when the energy state of the cell is low? Yeah, it will also increase. And why might that be? Yeah, exactly. You're lacking the ultimate product, which is going to be ATP. So you're going to want to make more by going through both of these processes. And which molecules might you have a lot of, indicating that you have a low energy state? Yeah. Yeah, ADP and AMP. And you might have a little bit or low amounts of what molecule? Yeah, ATP. Yeah. Yeah.
I have a question. So I thought you need a lot of energy in order for like PDC and stress stuff to happen. So I feel like it's kind of confusing to understand.
So none of them are requiring ATP as an input. So they are, during both of these processes, you're making reduced electron carriers. And in one of them, in the CAC, you actually do make one GTP, which is directly a source of energy currency. So both of these processes help you take not a lot of energy and make stuff that will help you make a lot more energy currency.
But then you need glucose as a reactant for this. Because with high levels of glucose, kind of like runs out of high levels.
So not always. So if you have a lot of glucose, you can go through these processes to make a lot of energy. But a lot of times, or we need to take in nutrients like glucose to help us make more energy currency. Yeah, and in the absence of glucose or other nutrients, it's very difficult to have high levels of energy, because that's like our whole thing as heterotrophs, right? We need to take in organic molecules like carbohydrates and use them to make things like ATP to help us go.
Wait, and then, but don't you need ATP to do like glycolysis?
You need a little bit to start, that's true. But ultimately, the glycolysis net yields 2 ATPs.
Oh,
So you use 2, but you make 4. So to get started, you do need a little bit. Yeah. If you had,
that glycolysis comes before these.
it does. Yeah.
So wouldn't you need to go through glycolysis in order to get through PDC and CAC?
Yes, because that is the source material for your pyruvate and your acetyl-CoA.
So there's no ATP you can get through glycolysis or pyruvate to get through.
That is true. If you had 0 ATP, you could not do it. But there's almost never a situation when you have 0 ATP molecules.
So you just, in this
Yeah.
case, you have enough to get through glycolysis.
Yeah. Yeah, it's like low versus high. It's kind of like everything else in biology. It's not, nothing is like absolute. Like you're never in a state of like 0 or like 100%. It's always like more of this or less of that, like increases and decreases, as opposed to like starting and stopping, if that helps. Okay. Yeah. Any other questions? All right. Yeah. And I guess to answer your question, if you had like 0 ATP, the cell would probably die, because like it just couldn't do anything. Okay, yeah. So let's go with the next one. So what happens when NADH starts to accumulate? Do we have increase or decrease flux? Yeah, decrease flux. Yeah, and why would that be? Yeah, exactly. So a product is accumulating. So when your product accumulates, that means the forward reaction is going to go more slowly. And why would NADH start to accumulate? What is missing from the environment if NADH is accumulating? Yeah, oxygen. Yeah, which brings us to the last question. What happens when oxygen is scarce? also decrease. Yeah, for the same reason, right? The products are accumulating. So that's going to slow the whole process down. And essentially, when we start talking about the electron transport chain, it's sort of going to unbalance the equation that we need for the electron transport chain. So like in a very simple way, we'll talk about it more on Thursday, but for the electron transport chain, we're taking NADH, we're using oxygen as the electron acceptor. NADH is finally getting oxidized, and oxygen's getting reduced to water. And through this process, we're making ATP from it. And we'll talk more about that on Thursday. But so you can think of, if there's very little oxygen, this forward reaction can't proceed. And so, yeah, so that's really the whole reason why we as organisms need oxygen, to help us make lots of ATP. All right, any questions about citric acid cycle, pyruvate dehydrogenase complex? Okay, so yeah, so just again, I told you at the beginning of class, but just as a reminder, quiz 12, if you haven't done it yet, or if you're going to do it, it's due this coming Thursday night. Your projects are due this Friday night. Your exam 3 will be on 2 Thursdays on the 14th of December. So Thursday, we'll be going over chapter 18, which is the electron transport chain and oxidative phosphorylation. And then that's it, believe it or not. So again, like I mentioned, if you want to see me about anything, I have office hours. My office hours are a little bit different the next couple of weeks, so check on Brightspace for my updated office hours. You can always send me an email. And yeah, that's all I got for you today. Have a great rest of your day.